Every year, hundreds of scientific papers describe exciting new stem cell-derived models, organoids, co-cultures, new differentiation protocols, and increasingly sophisticated human in vitro systems. Yet only a handful of those assays become ones that pharmaceutical companies routinely use or that regulators are willing to consider when making drug development decisions. The question is why? The answer isn't simply better biology. It's predictivity, reproducibility, standardization, and demonstrating that that model is fit for its intended purpose. Ultimately, the value of a stem cell-derived model isn't determined by how closely it resembles human tissue in the laboratory. It's determined by whether it generates reliable, interpretable data that can support a real decision. And now we're seeing some human stem cell-derived assays make exactly that transition, from promising research models to technologies that influence drug discovery and safety assessment. I'd like to illustrate that using two examples I've been fortunate to work on. The first is the human iPSC cardiomyocyte MEA assay for assessing proarrhythmia risk, or in other words, it's the risk that a drug could trigger a dangerous abnormal heart rhythm. The second is the emerging human iPSC neural MEA assay for seizure liability. They're at different stages of maturity, but together they illustrate how a promising model can evolve into a trusted NAM. And by NAM, I mean a new approach methodology, broadly a technology or approach designed to answer a specific hazard or risk assessment question more efficiently than existing methods. The key is that a NAM should be fit for purpose, able to generate relevant, interpretable evidence for the decision being made. For human safety assessment, that often means improving our ability to predict human biology and understand the mechanism. And when a NAM does that, it can also reduce reliance on traditional animal testing. For example, by identifying high-risk compounds earlier, reducing repeat studies, or supporting decisions where animal models are less relevant. And regulatory thinking has evolved alongside the science. This shift didn't suddenly begin with recent legislation. Regulatory engagement with NAMs has been building for more than a decade through initiatives such as SIPA and FDA-led validation studies. The FDA Modernization Act 2.0 provided additional flexibility around how non-clinical evidence can be generated. But the key point is that the scientific bar hasn't been lowered. Regulators still need confidence that an assay is relevant, reproducible, and appropriate for its proposed context of use. So what does that look like in practice? Cardiac safety provides perhaps one of the clearest examples. A major concern is whether a drug could provoke a dangerous arrhythmia, particularly torsade de pointes, or TDP, a rare but potentially fatal ventricular arrhythmia. TDP is associated with prolongation of the QT interval, which reflects delayed electrical recovery or repolarization of the ventricles. Because QT prolongation can signal increased TDP risk, it became a key focus of ICH S7B and E14 guidelines. The S7B non-clinical guidance relies heavily on hERG potassium channel inhibition, which can delay repolarization and prolong the QT interval, together with QT measurements in animal studies, typically in beagle dogs. Those approaches have been very effective at protecting patients, but they also have limitations because they focus largely on surrogate markers of risk rather than measuring the integrated human cardiac electrophysiology that ultimately determines whether arrhythmia occurs. Human iPSC-derived cardiomyocytes offer a potential solution. Rather than measuring a single ion channel, these cells incorporate multiple interacting ion channels within a human cardiomyocyte, so we're moving towards an integrated functional system. When these cardiomyocytes are cultured on multi-electrode arrays or MEAs, we can non-invasively record their electrical activity. We can measure parameters such as field potential duration, beat rate, conduction, and rhythm. And importantly, we can detect arrhythmia-like behavior directly. That's the distinction I want to emphasize. The assay measures function.